Hi everyone, I'm Caitlin,
And I'm Hannah.
and for our enzyme webinar, we chose succinate dehydrogenase.
To start, we're talking about the basic structure of the enzyme succinate dehydrogenase, which is shown in the image on the right. This image was taken from Proteopedia, a bioinformatics website, which uses JMOL for their animations and images. The basic structure to start, it has secondary structures of alpha helices and anti-parallel beta sheets. They're a little bit hard to see in the image, but you are able to see them if you look a little bit closer. It is a tetramer with succinate dehydrogenase subunit A, shown in blue, SDHB, shown in green, SDHC, shown in pink, and SDHD, shown in yellow.
And succinate dehydrogenase also has two binding sites. They're a little bit hard to see, but they're the gray and red and blue balls in between the subunits. And so there's one binding site specific for succinate and one specific to ubiquinone. In terms of residues and stabilization, there are several amino acids throughout the enzyme that are found within each of the four subunits that help to specifically stabilize the substrates. And so this picture on the right-hand side is also taken from JSmall via Proteopedia, and it's zoomed in specifically on 3 of the important amino acid residues within SDHA that are helping to stabilize the substrate. So we can see here that they are arginine, histidine, and threonine, which, like I said, help with hydrogen bonding. They form hydrogen bonds that allow for the substrate to be stabilized. And also, as with any tertiary structure, there are hydrophobic interactions and electrostatic interactions that help stabilize the structure.
This slide is important in understanding the function and also the location of the enzyme. In prokaryotic cells, it's located in the cell membrane, and in eukaryotic cells, it's located in the inner mitochondrial membrane as shown in the image. Starting from the bottom, the citric acid cycle converts succinate to fumarate via oxidation with the help of the cofactor FAD in subunit A. FAD is converted to FADH2. Subunit B has 2 prosthetic groups. One of these is an iron sulfur complex, and the other is the heme B group. And there is a space in between subunit B, C, and D that is the binding region for ubiquinone, where ubiquinone is converted to ubiquinol via metal ion catalysis. The electrons from that reaction are then converted and are then moved to complex 3, where the electron transfer chain can take place. It's a cool fact about this enzyme is that it is one of the only ones involved in the citric acid cycle and the electron transfer chain. Also, it is important to note that subunits C and D are hydrophobic subunits, and the other 2 are hydrophilic. So their hydrophobicity allows the enzyme to be embedded in the inner mitochondrial membrane for its location.
So like Hannah said, subunit SDHA helps promote the oxidation of succinate to fumarate. So this is specifically an elimination mechanism as well as an oxidation reaction. So what's happening is that there are 2 hydrogen atoms on succinate that are removed, and they're given to FAD to then produce FADH2. And so then because those 2 hydrogen atoms are lost, it promotes the formation of the double bond as with a typical elimination mechanism, and that results in our product fumarate. And so as with any oxidation reaction, we have both the loss of those H+ atoms, which are given to FAD, and also the loss of electrons. And so those 2 electrons that are lost in the reaction will then be used to reduce ubiquinone. So as for the reduction of ubiquinone to ubiquinol, ubiquinone has the -one suffix, which indicates that there are ketone groups on the molecule, and it's being reduced to ubiquinol, which that -ol suffix means that there is alcohol groups on the molecule. So what's happening is with this reduction reaction, we're gaining hydrogen atoms, and so those ketone groups are then being reduced to alcohol groups. And as we can see how they each now have an H on them. And so then those two electrons are then transported via subunit C and D, or with the help of their anchorage, to complex 3, also known as the electron transport chain, where they can then be used to help generate ATP. And so in summary, the oxidation of succinate to fumarate and the reduction of ubiquinone to ubiquinol verifies succinate dehydrogenase as specifically an oxidoreductase enzyme. So also, succinate dehydrogenase is unique in the fact that there are 2 different types of competitive inhibitors that the enzyme can have. So there's succinate analogs, which specifically bode structural similarity to succinate, and then there's also ubiquinone analogs, which bode similar structure to ubiquinone. And so what happens is those succinate analogs, for example, would bind to the succinate binding site instead of succinate, and therefore inhibit the binding of succinate, which would also have an effect on the Km of the reaction, as we learned about for competitive inhibitors. And so in this slide specifically, it's showing malonate as an example of a competitive inhibitor, as it has a similar structure to succinate, and thus hypothetically malonate would bind to the binding site of succinate instead of succinate, and that would prevent the oxidation of succinate to fumarate.
Okay, for our primary source, we decided to analyze a paper that investigates the effects of various mutations in subunit C on enzyme activity. We're aspiring to remember that subunit C plays a large role in the reduction of ubiquinone to ubiquinol, as we've been talking about. The first amino acid sequence in this figure is the full-length isoform, which is 169 amino acids long. It has the start codon of ATG. It includes all 6 exons. It includes the heme binding region and stops at a stop codon. The one right below this is the delta 3 ASV, which means that it is an alternative splicing variant with the loss of exon 3. As we can see, that would include a 102 nucleotide deletion shown in the gray box. It still starts with the start codon of ATG and still stops with that same stop codon. In the final amino acid sequence in this figure, it is a delta 5 ASV, meaning it loses exon 5. It still starts with that same start codon. It includes the first 4 exons, but there was 164 nucleotide deletion of the entire exon 5. Because it is 164 nucleotides, that's not a multiple of 3, so it shifts the frame. It's called a frame shift mutation. So it's not, the amino acid doesn't stop where the other stop codon was located. There are 70 more nucleotides that are read past that point until it reaches its stop codon. Additionally, it's important to note especially that the heme binding region is lost with this delta 5 ASV, which means that there will be electrons left in the inner membrane space.
As for the methods to start, RNA was extracted from normal lung tissue so that there could be a control group of normal cells that the experimental group can be compared to. And then secondly, the HCT15 cells, also known as colorectal cancer cells, they were treated with 10% heat inactivated fetal bovine serum, and they were also then grown under standard cell culture conditions in order to grow those cells so that they would be able to extract the RNA to then compare to those of the normal cells.
Mm-hmm. The RNA underwent cDNA preparation so that reverse transcriptase PCR amplification could occur. In order for that amplification to occur, the researchers had to design primers that would yield full-length delta 3 and delta 5 isoforms, all individually. Once these were all amplified, they were separated based on weight via gel electrophoresis.
The graph on the right-hand side of the slide is displaying the relative value of SDH activity. So essentially, the amount of SDH activity versus each of the 3 different amino acid sequences. So the full-length isoform, delta 3 ASV, as well as delta 5 ASV. And so from the bar graphs, we can see that the full-length isoform demonstrated the highest level of SDH activity, followed by the delta 3 ASV. And then finally, delta 5 ASV demonstrated significantly less SDH activity than the other 2. And so the asterisks that are directly on top of those 3 bar graphs, those are in reference to p-values, specifically to the p-values, or specifically to the normal cells. So the p-value, if there's 1 asterisk above that bar graph, that means that there's a p-value of less than 0.05. And if there's 2 asterisks, that indicates a p-value of less than 0.01, both of which are statistically significant, but a p-value of 0.01 is even more statistically significant. So what those asterisks right above the bar graphs are saying is that there is statistical difference in each of the 3 amino acid sequences, the full length, the delta 3, and the delta 5, in comparison to the normal cells. So in other words, there's significant difference in SDH activity for all 3 of them in comparison to the normal cells. And so also then the lines in between each of the bar graphs that also show 2 asterisks, those are saying that there's p-values of less than 0.01 between the full-length isoform, the delta 3 ASV, and the delta 5 ASV. So what that is saying is the results are statistically significant between each of those 3 amino acid sequences. So there's significant difference in the SDH activity between the full-length isoform, the delta 3 ASV, as well as the delta 5 ASV. And so because those p-values are so low, we're able to reject the null hypothesis that essentially says that there is no difference in SDH activity between any of the 3 categories. And because we reject that null, we are in turn able to accept the alternative hypothesis that not only is there significant difference in SDH activity for those 3, but the delta 5 ASV in particular also acts as a dominant negative inhibitor.
Like Caitlin's saying, it's super important to recognize the statistical significance, ultimately because it leads to the 2 main conclusions of the results of this primary source. The first is that delta 5 ASV is a dominant negative inhibitor. This means that because the delta 5 ASV because SDH activity decreases with that variant, even with the presence of a full-length isoform that increases SDH activity, it doesn't matter because that delta 5 ASV will dominate. Also, the second conclusion is that there is superoxide formation. Like I mentioned before, there will be electrons left in the intermembrane space with the delta 5 ASV, because ubiquinone cannot be transferred to ubiquinol when there's that mutation in subunit C. So the electrons will bind to that electronegative oxygen that's in that space, creating a superoxide. Although we are still investigating the specific effects of superoxides, we know that we do know some things about them. One of them is that it alters the expression of cancer-related genes, which can form tumors that are cancerous, and that's obviously not good for your health. And they also have other toxic effects on the cells that can lead to dysfunction of the cells. So in total, in conclusion, not only does this variation, this alternative splicing, with the removal of exon 5, alter the overall function and stop the function of succinate dehydrogenase from happening, but it can also have detrimental health effects. Finally, here are our references. Thank you for listening.
Thank you.
How long was that?